It gives me great pleasure to introduce our initial speaker because as you're going to see, we're going to begin now with a brilliant scientist that will present his research of quite a number of years on this particular topic. I've had the great pleasure of lecturing with Dr. Ritchie, and I can tell you that you're in for a treat as a kickoff speaker for this morning because he will lay the foundation for what is to follow. Dr. Ritchie is an associate professor in the Department of Biomaterials and Biomimetics at NYU College of Dentistry. He graduated from Muhlenberg College in Pennsylvania in 1977 with a Bachelor of Science degree and subsequently earned his PhD from UMDNJ in '84. He has a number of research interests, one of which is the development of controlled microtexture surfaces for dental implants that promote bone, connective tissue, and epithelial attachment. He is one of the developers of the Laser-Lok microtexture surface. He is also working on the development of three-dimensionally printed scaffolds consisting of permanent and resorbable materials, another exciting topic down the road, another great technology for replacement and regeneration of craniofacial bone and soft tissue structures. He is well published, and I can tell you that this is a gentleman that brings very esoteric scientific material and distills it in such a manner that we can all understand it. It gives me great pleasure to introduce Dr. Jack Ritchie.
Thank you very much, Mike. I'm very excited and very proud to be here today. Also a little bit humbled because as a basic scientist, it's not often that you get a chance to do this. I was trained really as a cell biologist, anatomist, and materials scientist. And most of the time, when you're a researcher like I am, you don't get a chance to see your projects actually turn into something real that's a real product. And this is something that I've been working on literally for 20 years. So this is really a lot of fun for me today. The only thing that's a little scary about it is the people I'm up here with Mike Picos and Myron Nevins and Stu Fromm. It's like, it's a little bit scary company to be here with. Anyway, what I'm going to start with today is literally the science behind the Laser-Lok microchannel surface. This is a project that started an awfully long time ago, and it started the right way, and I'm going to show you a little bit how things start the wrong way. This comes to me by John Brunsky, who lectures for a master's program that we do in biomaterial science, and I thought it was appropriate. The way we're supposed to develop biomedical devices and the way we teach our students that it's supposed to be done is you're supposed to start with a clinical problem, and you're supposed to do some engineering and prototype development. And there's a logical progression that's supposed to happen in this process. And you're supposed to do animal studies and refine the product, and then go through a clinical trial, and then marketing, and then application eventually. And unfortunately, with dental implant surfaces, we haven't always followed that plan. And most of the things that are on the market, and I'm being a little cynical about this, are essentially marketing-driven surfaces. We find something that's a cool-looking surface. Then we find a way to mass produce it, put it on an implant. We make lots of them and get FDA approval. And getting FDA approval for an implant is not that hard as long as you're not trying to get any claims from the FDA, at least here. You sell a lot of them. And then eventually, you do some animal studies so you can back into some science to support your marketing. And you end with a clinical trial that shows at least a 95% success rate. And this is a rule that is present pretty much in dentistry as well as orthopedics. I spent 16 years in the orthopedics field, and there's some rules that cross the lines between both fields. And one of them is that, in general, you don't publish too many clinical trials that show less than a 95% success rate. You occasionally see a 93 or something like that. But unfortunately, that's the way things have worked in this field, which is really not very science-driven. And what it's led to is some rather interesting designs and some things that we've learned a lot from. I used to run the implant retrieval lab at Hospital for Joint Diseases when I was in orthopedics. So we used to see all the interesting things that fell out of people for one reason or another, and you can learn an awful lot from your mistakes. And we saw a lot of mistakes. And actually, a few of them are here. And some of this stuff just, there wasn't very good science behind some of it. This is actually from an '88 cover from the Journal of the California Dental Association. Even in 1988, there were so many different implants around with, different thread designs and different concepts, cylinders, HA-coated cylinders, single crystal sapphire implants, all kinds of wild stuff that was around. And very little of it has survived the test of time. Certainly some of the implants were interesting concepts. The Zircon implant, what we call the Molly Bolt implant, the old blade implants were an interesting design, and they were the beginning of the current implants, but they've been replaced too because they're a little too surgically intensive and they didn't last very long sometimes. This one's one of my favorites because this one kind of comes out of the orthopedics field. In orthopedics, when you do intramedullary rods, they very often use a fluoroscope to put pins in the ends of them to fix them. And this is a dental implant that had a fixture that fit on the collar that allowed you, it had a drill guide, so you could put pins in to hold it in place. The only problem is it was a little bit cumbersome trying to fit all that in the patient's mouth, and it was a little bit surgically invasive, to say the least. The way we developed Laser-Lok was different, and it was because it started as a basic science project. What happened is we started with a clinical question in 1988. Basically, we said, how does texturing work? We knew that texturing produced a different response than non-textured surfaces. And so we started doing some very basic science studies to look at how cells respond to textured surfaces. We then started in the early 90s trying to transfer that technology, what we found, the surfaces we identified, onto metal and make metal prototypes. And we then began doing what are called animal screening studies. And when I say screening studies, I mean studies that are designed to test multiple surfaces and see which ones work the best. We then did the refinement part of that where we put the surface on a dental implant and we did what's called an animal application study where we tested them in animals. Then we started human clinical trials, and this was originally done with Biolok, which we started working with in 2000. The implant received FDA approval after clinical trials and the animal data in 2004, and then Biolok was acquired by BioHorizons. And BioHorizons has done more refinement on the surfaces, has produced some very interesting implant designs, and really what has come out of this is what I would call the next generation Laser-Lok implants, the next generation surfaces and the implants. And as you'll see, these are very interesting, and they're going to be very successful. What we started with is this implant on the left. This is the Biolok Silhouette implant. It has, you can see it's a tapered implant. It has an interesting thread design called a reverse buttress thread that puts compressive load on the bone. It has this tissue engineered collar that's mainly what I'm going to talk about today. That's the Laser-Lok collar. And it has what Biolok call an Osteolok surface, which is a high roughness, clean blasted surface. Now, what that has led to, of course, is the Laser-Lok tapered internal implant by BioHorizons, which you'll notice is very similar. The thread design is similar. The main difference is the collar portion of the implant now has BioHorizons internal connection. And other than that, the surfaces are the same. One of the interesting things about the acquisition of Biolok by BioHorizons was that the companies had very similar philosophies. It was a tremendous fit for Biolok to be part of BioHorizons because we even use the same surface. This surface